Hi, students. So let's go ahead and go over the key points from Module 4, which is all about DNA structure and function. So we are going to start by talking about some of the important discoveries that helped us figure out why DNA is so special. So you should remember from the first week of class that DNA is the genetic material. It's the hereditary information, meaning it is the molecule that is passed from parents to offspring. So originally, it turns out when scientists were learning about all those 4 different classes of macromolecules, that a lot of them thought it must be proteins that were passed on from parents to offspring. And that's because there are 20 different types of amino acids. And remember, amino acids are the monomers of proteins. But there's only 4 types of nucleotides. And remember, nucleotides are the monomers of DNA. And so at the time, they were like, well, proteins have 20 letters in their alphabet. So that must be a much more complex language. Therefore, that must be the genetic information. Like DNA, it only has 4 letters. Like that's just not enough information. So at the time, people thought DNA was just like this stupid molecule. So there's no way it could be the genetic information. But it turns out DNA is the genetic material. And the difference is that DNA as a molecule is just really, really, really long. It's much longer than any protein. So even though there's only 4 types of nucleotides, the words in the DNA language are just way longer than proteins. So that gives you that extra complexity. So for example, in humans, the longest protein is 35,000 amino acids, which is pretty humongous, but the longest DNA molecule is 249 million nucleotides. So that's what, like 10,000 times longer almost. So DNA, just because it is so long, it sort of makes up for the fact that it only has those 4 letters. So just something to keep in mind as we are thinking about how scientists discovered that DNA is the genetic material. They actually had to do some pretty robust work to prove to the rest of the scientific community that it was DNA that mattered and not proteins. So how did scientists do that? So the first scientist who was important in this was named Erwin Chargaff. And what Erwin Chargaff did is he was able to figure out how DNA base pairs with each other. So what that means is DNA exists as a double helix. So we're going to use this little model a lot today, the zipper, to represent DNA. So normally DNA is a double helix, meaning it's like twisted like this, but just for simplicity, this little zipper is straight. So one of the cool things about DNA is it can unzip and then it can rezip back up together. So what this guy, Chargaff, figured out is how these letters match up in order to create that zipper. So just like a real zipper, like the teeth have to fit together just right in order for it to zip up properly. Same thing is true with DNA. The nucleotides in DNA like to match up in a very specific way. And those rules for how the DNA nucleotides match up in those 2 strands so that they can form that zipper, Chargaff is the one who figured out those rules. We're not going to talk about how he did that. We are just going to talk about the fact that he figured it out. And then there's just going to be a little tiny smattering of math that you're going to have to do here. It's not bad. So these are what Chargaff figured out. He figured out that one, by looking at a bunch of different DNA molecules from many, many, many different types of animals and plants and bacteria and everything, he saw that it was always true that the percent of adenine in one molecule always equaled the percent of thymine. So in other words, if a molecule was 25% adenine, it was always going to be 25% thymine. And because of that, he figured out, oh, A must always base pair with T. They must be together. That's why the amount of A equals the amount of T, because they're equal. So he figured out the amount of A equals the amount of T, and then the amount of G equals the amount of C, because there's only 4 DNA nucleotides. So if A equals T, then G must equal C. And then the next rule, this one is like for math reasons. If it bothers you, I can like show you the proof for this. It's not too crazy, but it's just like you don't need to know it. So just memorize in your brain that A equals T, G equals C, and then A plus G is half, and T plus C is half. It's 50%. So in other words, if it's not like A to T, if it's some other combo, it's probably going to be half. Okay, so those are those three rules. I should have mentioned this at the start. You do have a handout for Module 4. You should be filling that out as you're doing this, and hopefully you have those rules in that handout because we are going to move on to a check for understanding. Obviously, instead of doing this in the Pear Deck, you're just going to answer this question in your handout. It'll give you a second to think about it. So a DNA molecule contains 30% adenine. What percentage of the DNA will be thymine? So it's 30% A. What percent will be T? Okay, so if you go back and you look at those rules from the previous page, you'll see A equals T. So if it was 30% A, then it must be 30% T. So the answer is 30% because A is going to always equal T. Okay, so let's do one more. That same DNA molecule, 30% adenine. What percentage is going to be guanine? Okay, so this one you're going to use that second or the last rule, A plus G equals 50. This is that one where it's like, just trust me on the math, just memorize it. So if adenine was 30%, so then it would be 30 plus percent of G equals 50. We're going to solve for a percent of G, so we would get the percent of G would be 20%. So it's 20% guanine. Okay, that's all the math you have to do for this. It's not bad. That is our first scientist there, Erwin Chargaff. He's the one who helped us figure out those base pairing rules. So A goes with T, C goes with G. Okay, so then the next set of scientists who were very important in these DNA discoveries was two scientists named Hershey and Chase. And they were actually the ones who figured out that DNA was the genetic material and not proteins. Remember, I said before that most scientists at the time thought, oh my gosh, it has to be proteins because proteins are just like so much more complex than DNA. But Hershey and Chase proved that it was actually DNA. And how did they do that? So what they did is they used these special types of viruses called bacteriophages. So these are viruses that infect bacteria. That's why there's bacteria in the word. So here's a little picture of these little bacteriophages. They sort of look like little robot creatures. And the way these viruses work is they land on top of a bacteria and then they shoot their genetic material into the bacterium. So here's that little phage. It's shooting its material into the bacterium, and then you can see its genetic material is now inside of the bacterium. So they knew at the time that that's how these viruses worked and that they shot their genetic material into the bacterium, but just they didn't know what that genetic material was. So what they did was they took some of these viruses where they labeled all the proteins with a radioactive tracer. And then they had another version of the bacteria where they labeled all the DNA with a radioactive tracer. So what they saw was that in the bacteria that were mixed with the viruses with radio labeled DNA, that now the radioactivity was inside of the bacteria. Whereas in these guys, where the radioactivity was on the protein doesn't go inside the bacteria. The protein just floated away. So none of these bacteria would become radioactive. So that's how they proved that it must be the DNA that was getting shot into those bacteria. And then what they actually did was they took those bacteria and grew them over time and like let them reproduce and make lots of bacteria babies. And they saw that that radioactivity was still there because that shows that it was the DNA getting passed from parents to offspring once that DNA was inside. Was inside of those bacteria. So once again, Hershey and Chase, they helped demonstrate that DNA was the genetic material, meaning it's what was passed from parents to offspring. And the way they did that was with these very special viruses called bacteriophages. Okay. And all the next scientists we're going to talk about, they're all the ones that helped us figure out that double helix shape of DNA. So remember, once again, here's our lovely little model of our two strands of DNA. And it's a helix or a double helix, meaning it's kind of twisted like this. So this is sort of what a DNA molecule looks like. It's a complex shape, and it took a lot of time for people to figure out what that shape was. So it was discovered by a team of scientists in London. Two people on that team, Watson and Crick, are often given a lot of this credit because they received a Nobel Prize for this work in the 60s. And what Watson and Crick did was they actually built the first accurate model of the shape of DNA. So they literally took like little pieces of cardboard and glue and like made a big accurate model to show like with very accurate distances and everything, like this is how DNA must actually look inside of a cell. I think that sounds like kind of silly to us now, but it was very revolutionary at the time, and it was the first time anyone had ever done something like that. So Watson and Crick get a lot of the credit. They would not have been able to build that model had it not been for the work of Rosalind Franklin. So she was a scientist at the same institution in London who took an X-ray picture of DNA, and here's the picture she took. And then she did some math that's like way beyond calculus, like calculus plus 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 plus. And using that crazy math, she like measured the distance between these black lines, and somehow using math figured out that the shape of DNA must be a double helix. It's crazy to me. That's like crazy smart. She did it all like by hand without computers. But so she was the one who crystallized those DNA crystals, and then she took that X-ray picture of DNA, and her picture was what was used to figure out that DNA was a double helix shape. And then Watson and Crick built the first accurate model of that double helix shape. Rosalind Franklin, because she worked with X-rays, actually died of ovarian cancer in 1958. And so because of that, she did not get awarded a Nobel Prize, because the Nobel Prize is not awarded to anyone after they have died. So because of that, she has been often like kind of forgotten by history, which is sad because she was an integral part of the team. But so those were the last groups of scientists you need to know about the important DNA discovery. So Watson and Crick built the first accurate model, and then Rosalind Franklin took that picture that helped show that DNA was a double helix. Go. We're ready. Uh-oh. There we go. Okay, so check for understanding. What important contribution did Rosalind Franklin make to the scientific discovery of the structure and function of DNA? Go ahead and answer this question on your own handout before I show you the answer. Okay, if you need more time, you can always pause the video. But here's what I said. So Rosalind Franklin took the first pictures of DNA to discover the double helix shape. Remember, she did that using X-ray, so it was like an X-ray picture. Okay, so those are all of the important scientists involved in helping us figure out the importance of DNA. So now we are going to go to the next topic, which is understanding the difference between DNA and the other nucleic acid polymer, which is RNA. Remember, there's only two types of nucleic acid polymers, DNA and RNA, and that's it. They don't have their own special names. So we are going to learn about the differences between those two. So regardless of the differences, they do have a lot of similarities. So they're both made of the same types of monomers, nucleotides, but DNA is made of DNA nucleotides and RNA is made of RNA nucleotides. So you can kind of think of it like they both are made from nucleotide letters, but they're in like slightly different fonts. So they're very, very similar nucleotides, just like little tiny differences that sort of matter when you're talking about DNA versus RNA. Okay, so we're going to start by talking about that DNA polymer first. So once again, here is a nice picture showing that double helix shape. If I use my zipper again and I twist it up, here's what that double helix would look like. In this picture, these like colorful lines that almost look like rungs on a ladder, those would be representing those nucleotide base pairs. Those are those rules that Shargaf figured out. A lot of times when we think about DNA, we think about it just like straightened, like we've flattened out that helix just because it's easier to look at that picture. But just keep that in mind as we're talking about all this DNA stuff. So once again, DNA acts as the genetic material that provides instructions to produce proteins. So it's genetic material, meaning it is what is passed from parents to offspring. And the reason that DNA is important is because it tells your cells how to make proteins. Turns out that every cell in your body has essentially the exact same DNA inside of its nucleus, which is crazy. That's because you came from one single cell. That one single cell was a sperm and an egg that joined together in fertilization. And then that one single cell became you, right? That cell had to divide hundreds, thousands of times to become a full baby during those 9 months of gestation. But all those cells that made up that baby came from one original cell. And because of that, in theory, all the cells in your body have essentially the same DNA. Like, yes, there's a couple of mistakes here and there, but essentially every single cell in your body has the exact same DNA inside of its nucleus, which is crazy. And then once again, DNA exists as two strands wound together. That's called a double helix. So that's why I like to think of this zipper analogy. So you can unzip a zipper, right? So when we unzip that DNA strand, those two strands from each other, so the double helix, and we sort of like make it into two single pieces, this part that we just unzipped, but normally holds those two strands together, like the zipper itself, those are weak interactions called hydrogen bonds. So it's nice that they're weak because we're able to easily break them when we unzip the DNA. And as we're going to learn about today, the ability to unzip the DNA is important for just how DNA works. And then, as we've learned multiple times by now, each strand of DNA itself is a polymer. It is made up of many nucleotide monomers. So nucleotides are the letters that make up the DNA words. Okay, check for understanding. What is the term for the weak attraction between base pairs that holds together the two strands of DNA? So like the two zippers. So those are hydrogen bonds. Those are those weak attractions that hold together those two strands. They are like the zipper that we can break and then reform. Or zip and unzip. I guess you don't break a zipper. Okay, so that's just DNA really quickly. Now moving on to some more detail about nucleotides, which once again, nucleotides are the monomers of nucleic acids. They are the building blocks, the letters that make up DNA. So there's 4 types of nucleotides. We've already been talking about them. They're called adenine, thymine, guanine, and cytosine. That's just the names of those 4 letters. For short, we'll just call them A, T, G, and C. You should recognize those words, but you don't need to like spell them. But you should be able to know, like, oh, if you see the word guanine, that is a nucleotide. And here are the chemical structures of each of these 4 nucleotides. I know it's a lot to look at, but we're going to break it down and hopefully it'll be a little bit less crazy. So let's look at this one up here first. This is guanine, G. So see how on the left there's 3 gray dots, and then there's this blue pentagon, and then there's this big yellow thing that says G. Okay, let's compare this to this one underneath, which is A. There's 3 gray dots, a blue pentagon, and then now a pink thing that says A. And to see how this pink thing, I mean, obviously it's pink, but it looks different. Like this has an NH2 at the top, and this has an O, and then like an NH2 over here. And then same thing. Here's T and C. Do you notice how the parts that are the same are the 3 gray dots and then the blue pentagon? And the only parts that are different are these colored bits up here. So what we just did, what we just kind of like broke that down, was we were looking at the 3 parts of a nucleotide. So this part here, those are called the phosphate groups. This blue thing is called the pentose sugar. And then this part is called the nitrogenous base, or the base. And so what makes each of the nucleotides different, so what makes each of these different monomers different from one another, is the base. Like this part's the same. The only part that's different is the colored part up top, the nitrogenous base. Because of that, sometimes instead of calling these nucleotides, we'll call them bases, because like the base is the part that matters. The base is the part that is different. And then also, since those nucleotides like to form pairs, sometimes we'll also call them base pairs. Those are all terms that essentially mean the same thing. So if I say nucleotide, or I say base, or I say base pair, what I'm talking about are monomers of DNA that are joining together to make a polymer. Okay, so that's DNA. That's one of the polymers of nucleic acids. We're just going to talk so briefly today about RNA and its structure. Next week in Module 5, we'll learn more about RNA and how, sort of like what it does. So what RNA is, once again, it's still a polymer of nucleic acids, but it's a little bit different because RNA is sort of like a temporary copy of one little piece of DNA. So remember in one of the previous slides, I said that your longest DNA molecule was like 230 million base pairs or something crazy. So it's really hard to like look at all 230 million of those base pairs at one time. So what RNA, the point of it is just taking like one little section of that DNA and making a little tiny copy of it And then that little tiny copy can like leave the nucleus and go do all the stuff it needs to do without having to deal with like all 230 million base pairs. Maybe you're only going to copy like 1,000. That's a lot more manageable. So RNA is just a temporary copy of a specific part of DNA. That little part of DNA that is copied is called a gene. So RNA is made inside of the nucleus of a eukaryotic cell, and then it leaves the nucleus and goes to the cytoplasm, where it's going to do things that we'll learn about next week. RNA itself, so DNA is a double strand. It's like 2 pieces of a zipper. RNA usually actually is only 1 single strand, so it'd be like half of a zipper. But it is still made of nucleotides. It's just made of RNA nucleotides, so like a slightly different font. So let's talk about what makes an RNA nucleotide different than a DNA nucleotide. There's still 4 different types of RNA nucleotides, but the difference between them is that they have different sugars. So if we go back to this picture, so these were the 4 DNA nucleotides. Remember, they all had the same 3 gray dots here. Those were the phosphate groups, and they all had the same blue pentagon. So if we were comparing DNA versus RNA, what is different between DNA versus RNA is the blue pentagon, actually. So that is shown in this picture. This is that same little pentagon. It's just all by itself and kind of has more detail on it. But here is that sugar. It's a pentose, pent meaning 5, so it looks like a pentagon. Here's the sugar in those RNA nucleotides. It's called ribose. And then here is that same little blue pentagon, but the one that's in the DNA version, which is called deoxyribose. This is sort of beyond the scope, but the reason it's called deoxyribose is because if you look here, this is just an H, but on ribose, it's an OH, so an oxygen and a hydrogen. So in DNA, there's 1 less oxygen here, so it's like deoxy. It's been deoxygenated. It's lost 1 oxygen. That's where that name comes from. So once again, all that makes those nucleotides different between RNA and DNA is the little pentagon, the sugar that is part of those nucleotides. In RNA, the sugar is called ribose, and in DNA, the sugar is called deoxyribose. That is actually what the R stands for in RNA, R for ribose, nucleic acid, and then D for deoxyribose, nucleic acid. Okay, so that's the difference between RNA versus DNA nucleotides. And then there is one other little difference. We're going to talk more about this next week, but as we learned earlier, there are 4 different types of DNA nucleotides, 4 different letters in the DNA alphabet. There's C, T, C, A, and G. In RNA, it's almost the exact same, except instead of T, there's a slightly different version called U, uracil. So it's U, C, A, and G. So RNA has this one special nitrogenous base called uracil, which is only found in RNA. So there's no T. We'll talk about this more next week, like I said, but just wanted to introduce this a little bit today. Okay, so check for understanding. Go ahead and answer this question in your packet, pause it so that you have enough time to write out your answer. But tell me what is at least one difference in the structure of DNA versus RNA. Go ahead and pause the video. Okay, there's lots of things you could have said here, but here's 3 examples. DNA is a double helix, but RNA is single-stranded. DNA has deoxyribose as its little pentose sugar, that blue pentagon, but RNA has ribose. DNA nucleotides are A, T, C, and G, but RNA nucleotides are A, U, C, and G. All good things that you could have said. Okay, so that is the difference between DNA and RNA. Oh, my animation is not work there. And then the last thing we're going to learn about is just those base pairing rules. We've actually kind of already learned this because these are those rules that Chargaff figured out. So Chargaff figured out that the amount of C equals the amount of G, and the amount of T equals the amount of A. So he is the one who figured out these base pairing rules. So in other words, if we look at our little zipper here, if on one side of the DNA strand it's G, the other side of the zipper, like the part that's going to match up to it, is going to be a C. If one part is A, on the other side, the strand that matches up to that, it's going to be a T. So those are those rules. So C goes to G, A goes to T, vice versa. Here's a trick to help you remember this. It's silly, but you can remember apple in the tree, car in the garage. So apple in the tree, A and T, car in the garage, C and G. Or honestly, the way I always remembered it is that the letters C and G look very similar. So I just was like, oh, they go together because C looks like G. And those are your base pairing rules for DNA. So let's do a little practice problem to see if you understand how to use this. Okay, so here is a sequence of DNA. It's CCGTAT. Go ahead and write in your packet what would be, so like if that's one half of the zipper, tell me what would be the other half of the zipper that would be complementary to this DNA sequence. So go ahead and pause the video and see if you can figure that out. Okay, so to do this, you have to use those base pairing rules. So what binds with C? Well, that's going to be G. So C goes to G. What binds with G? C. What binds with T? A. A goes with T, and then T goes with A. So that's your answer. We just kind of essentially, I had it like this, but you're just figuring out, or this, like the base pairing rules along the zipper. Okay, so that's sort of the basics of the structure of DNA. So our next big topic for this module is chromosomes. So chromosomes are a way to organize your DNA. So DNA, super duper long. Remember we said earlier that the longest strand of DNA you have in your cells is about 230 million base pairs long. That's crazy long. Each piece or each molecule of DNA does a lot of things. And within that molecule of DNA, there's going to be sections that code for specific products, meaning they're the instructions to tell cells how to make a specific thing. That's called a gene. So a gene is just a section of DNA that codes for a specific product. A way to think about it is like if your DNA is like a cookbook that has a lot of information in it, right? A gene would be like one single recipe inside of that cookbook that is telling you how to make one single thing, one single dish. Usually that dish is a protein because proteins are the only thing that matter, right? So as I said before, it turns out all cells in your body have the same DNA in them, right? Because you came from one single cell. So that means that all cells in your body actually have the exact same genes. But what makes different cells different is which genes are turned on and actively producing proteins. So put another way, like the DNA in your skin cells is the same as the DNA in like your liver cells. But what makes a skin cell different from a liver cell are the genes that are turned on. So like which recipes are being made from the same cookbook. That's what makes each type of cell different. You have a ton of DNA in each cell, like a crazy amount. I already said the longest piece is 230 million base pairs, but that's only one piece, and you have multiple pieces of DNA in each cell. So keeping track of all of that DNA and keeping it organized and making sure your cells know like which gene should be on at the right time is actually really, really complex and hard work for your cells. So that's why you need chromosomes. So all of the DNA in your cell is organized into these structures called chromosomes. So we're gonna, I hope the volume works. Fair sound. Does now, great. So we're gonna watch probably my favorite video ever about this because it just helps you remember like how much DNA is in your cells and why it is so important to keep it all organized into chromosomes.
You've probably seen DNA represented like this, but it actually looks a little more like this. It's an incredibly long molecule that's precisely folded and tightly packaged into the 46 chromosomes in our body's cells. How long exactly? Well, if you uncoiled all of the DNA in just one cell, it would stretch over 6 feet, and that's just in one cell. We don't know for sure, but recent studies have estimated the average adult body is made up of around 30 trillion cells. So if you lined up all of the DNA molecules in all of the cells of your body from end to end, they would reach over 34 billion miles. Now let's try to comprehend that distance. At their closest, the Earth and Pluto are 2.6 billion miles apart. NASA's New Horizons spacecraft, one of the fastest man-made objects of all time, travels around 1 million miles every day and still took almost 10 years to reach Pluto. Now take that distance and double it. Then double that. Then triple that. And you still haven't matched the length of all of the DNA that's tucked away in you right now, guiding your body's growth and repair.
Is that not the craziest thing you've ever heard? That you have inside of your body so much DNA. How do I get out of this video? No, don't play it again. Stop. Okay, so inside of each cell, one single cell contains about 6 feet worth of DNA. So think of someone around you in your life who is 6 feet tall, one of them of DNA, which is a crazy amount of DNA to start with. And then you have, what did he say, like 300 trillion, 30 trillion cells or something. So if you took all that DNA and you ended or like lined it up end to end, it would go from the Earth to Pluto 12 times. That's how much DNA is in your cells. That is crazy. That's why it has to all be organized into chromosomes, because you have so much DNA. It's just like, how could you ever possibly manage that? Sorry, I'm going to re-share this so that it's big again. Great. No. So that's why DNA has to be organized into chromosomes. That is why chromosomes are so important. What the heck are chromosomes? Let's talk about it. So here's a nice little picture to kind of break this down for you. So here's that DNA double helix. Remember, this picture right here, this part of it would be like looking at the little zipper, the zipper part that we're used to seeing. And then here it's showing the double helix shape. So we've twisted that zipper up. And then we take that DNA helix and we kind of like wrap it up around these little balls and then crunch it up even more so it gets really, really tiny. And that is a chromosome. So it's just that DNA that's been wrapped up really tight into a nice little organized package. So those little balls that the DNA wrapped around, those are something called histones. You can sort of think of a histone as like a spool of thread. So if you keep that thread organized and like wrapped around a spool, it's going to be a lot just neater and tidier and a more efficient use of space. And then that DNA that's been wrapped around that histone protein and all like condensed, that is called a chromosome. So we would say that in terms of like levels of organization, that a chromosome has a much higher level of organization than DNA, because a chromosome is DNA that's wrapped around a histone. So it's more organized. So it's a high level of organization. Most humans have 46 chromosomes in each of their cells. So once again, that comes from that one single cell that made you. So the sperm brought 23 chromosomes, the egg brought 23 chromosomes. They joined together to make a fertilized egg, and that fertilized egg usually has 46 chromosomes. And then those same 46 chromosomes get passed on to all of the cells that go on to make your whole body. So check for understanding, what is a gene? Okay, so hopefully you said something like a segment of DNA that codes for a protein. Next one, which has a higher level of organization, DNA or chromosome? So a higher level of organization, meaning it's like more organized, that's a chromosome, because a chromosome is DNA that's wrapped around those histone spools, so it's more organized. It's like more complex than just DNA. No. Okay, so now I have to try to minimize this again. Oh. Okay, so that's chromosomes. There are different types of chromosomes. There's essentially 2 different categories of chromosomes in each of your cells. So there are sex chromosomes. Sex chromosomes are the chromosomes that are involved in sex determination, meaning they are the chromosomes involved in phenotypic male versus female sex. Those chromosomes are in every single cell in your body, even though that's talking about sex determination. So you would think like, oh, ovaries, testicles, things like that. Those sex chromosomes are in every single cell, because once again, every single cell essentially has the same starting set of chromosomes. It's just which chromosomes and which genes are turned on. So you have your sex chromosomes, And then all of the other chromosomes are just called autosomes. Those are like all of the not sex chromosomes. They're just everything else. So once again, most humans have 46 total chromosomes, and that includes 2 sex chromosomes, and then all the rest, so 44, are autosomes. So this is like a little schematic showing what those 46 chromosomes could look like in someone. So this person has these 2 sex chromosomes called X and Y, and then everything else in this picture, these are all called autosomes, and they would have 44 of those autosomes. Okay, so let's talk about sex chromosomes more a little bit. We will kind of get into this more in next week or module 6. In the future, we'll talk a little bit more about sex chromosomes, but just to introduce this here, once again, sex chromosomes help to determine biological sex. So biological sex is the trait that determines what kind of gametes that individual will produce. So in other words, is that individual producing sperm, if it's an animal, or are they producing eggs? So in animals, sperm are like the mobile gametes that have the little flagella, the little tails. So a person who is producing sperm would have male, like phenotypic biological sex. In other organisms, like in plants, instead of having sperm, because remember plants don't have flagella, plant cells, they don't have sperm, instead they have pollen. So pollen is still a mobile gamete, it's the one that moves around, but instead of moving around with little tails, pollen is just moved around by the wind. So a plant that makes pollen would be a male plant, and then a plant that makes eggs is a female plant. Then we are kind of already looked at this picture on this slide where I was talking about, here's like a little depiction of the chromosomes. So this picture is something called a karyotype. So karyotype is really just a tool to help us visualize the chromosomes in a single cell. So this is what it would actually look like. So here's an actual microscope picture of one single cell right here in green. If you zoomed in on it, here's what it would look like. And all these little black squiggles, those are all chromosomes. So this is what it actually looks like in the cell. It's not like super organized. A karyotype would look like this. So what this is we took the same picture and someone just like kind of used Photoshop to like cut out this specific chromosome and like paste it over here. And that is what we call a karyotype. And just by convention, the way that we line or we make these karyotypes is we will organize it as longest chromosome to shortest. So the longest chromosome is by definition chromosome 1. And then the shortest chromosomes are by definition chromosome 22. And you'll notice that they always come in pairs. That's because one of those chromosomes came from or was carried by the sperm that created this person, and the other one was carried by the egg. And then at the very end, these last 2 chromosomes, those would be the sex chromosomes. Just once again, by convention, we just put those at the end. Oh, I said that. Yeah, that's what I said. So a karyotype, once again, it's like a laboratory tool that's used to help us visualize the chromosomes. This is not how your chromosomes, they're not all organized like this inside of a cell. They look like this. But there's very helpful tools, karyotypes, to help us understand if someone has like chromosomal abnormalities, for example. So once again, all the autosomes, those are going to be these first 22 pairs. So the first 44 chromosomes in a karyotype. And then these last 2, this last pair are always going to be the sex chromosomes. Just by convention, we put those last. And these are going to help us figure out if this person has male or female biological sex. So sex chromosomes in people, this is how biological sex is usually determined. Turns out sex determination is like super complex. For me, it's like a very interesting topic and something I could talk about a lot. But just as something to keep in mind is that sex determination for different species can work very differently, because this is a nursing school, like we focus on humans, but sex determination can actually happen in a lot of different ways in different types of animals. So for example, in a lot of reptiles, sex is determined by the temperature of the environment when the eggs are laid. So for example, in sea turtles, if it's really hot when those eggs are being hatched, those turtles are more likely going to be female, where if it's cold, they're more likely going to be male. In a perfect world, you want like the Goldilocks temperature so that you get about 50-50 male and female. But turns out as the temperature increases because of climate change, we're kind of starting to like get to this warmer part of the curve. So there's starting to be more female sea turtles than male sea turtles being born or being hatched. And that just makes it hard for the male sea turtles to find mates and then make more baby sea turtles. And sea turtles already have it hard. So it's rough times to be a sea turtle. But that's one example. Another example of really interesting sex determination is in some animals like clownfish, sex is determined by social factors in the environment. So you might have heard maybe that in Finding Nemo, when Finding Nemo's mom died, what would have actually happened is Finding Nemo's dad would have become the most dominant fish and actually would have switched to become female. So all clownfish have sort of like the ability to be male or female, and sort of like the biggest, most dominant fish in the school becomes the dominant female who will produce eggs. And then all of the other fish are either male or undifferentiated. So if the dominant female dies, then another fish will become the dominant female. So cool. So that's in other animals, but even in humans, it's a little more complicated. But if we want to try to simplify it, we would say that in humans and actually in most mammals, sex is usually determined by those pairs of sex chromosomes. So for us, it doesn't have to do with like the temperature or social cues, it's sex chromosomes. So most females have 2 X chromosomes. So the sex chromosomes for most female humans would be XX. And then for most males, the sex chromosomes would be XY. So hopefully you can notice this is a little screenshot from the karyotype. So for the X chromosome, it's much longer than the Y chromosome. So it turns out that there are a lot of genes and instructions on the X chromosomes that are important for life. Like a person cannot be born without at least 1 X chromosome. You have to have at least 1. But the Y chromosome, all it really does is tell that person to make testicles and sperm. So you can be perfectly fine if you don't have a Y chromosome. In fact, most of us do not have Y chromosome. All the people who are XX don't have one. Okay, and then even then, you might have noticed I kept saying most about sex chromosomes, even when talking about people. And that's because there are some very interesting chromosomal abnormalities that can happen that lead to just some more rare types of sex chromosomes. So it's estimated that about 1 in 500 males are XXY. So they actually have an extra X chromosome. This is called Klinefelter syndrome. This is a cat who has Klinefelter syndrome. Turns out that all calico cats, so calico cats are the ones that have like the orange and the black spots. Most of those cats are female because what makes that calico patterning is having 2 X chromosomes. But this cat is actually male. That has male genitalia, but it's also calico. The only way you can have calico is if you have 2 X chromosomes. And the only way you can have male genitalia usually is if you have a Y chromosome. So that leads you to believe like, oh, this is an XXY kitten. It's Klinefelter syndrome. So it's a very rare kitty. Very sad I didn't get to adopt this adorable little Klinefelter kitty. So cute. And then it's estimated that about 1 in 1,000 females are XXX, so triple X syndrome. They just have an extra X chromosome. Usually that doesn't hurt or harm you. You might not even know if you had an extra one. And then there's other interesting examples too that we're not going into, but another kind of more rare sex determination phenotype is it's estimated about 1 in 80,000 phenotypic females are XY. So they have an X and a Y chromosome. So we would expect that person to be female. So this is something called Swyer syndrome. So what happens is essentially the default for all fetuses is to become female. But then the Y chromosome, if someone has a Y chromosome, it tells the fetus like, hey, just kidding, start developing male sex characteristics, like start making testes. So in people with this syndrome, they have that Y chromosome, but it's defective. So because it's defective, they stay on the default female path, even though they have a Y chromosome. So this is why I keep saying most about sex chromosomes. Like, yes, usually XX is female, XY is male, but not always. And being that you guys are all going into healthcare, I'm sure that you also are interested in sort of the different types of sex determination and just the different sort of disorders that could be associated with different types of sex determination. Okay, so that's sex chromosomes. Let's do a quick check for understanding. In mammals, what are the sex chromosomes found in most males? Yeah, so most males are going to be XY. In terms of what you need to know for the final, you should know that. Most males are XY, most females are XX. Okay, so that is sex determination. We're almost done. So now we're going to talk about DNA replication. So DNA replication, oh no, where's my little zipper? So DNA is our lovely little double helix, right? So whenever a cell divides and makes another cell, what it has to do is copy all of its DNA. So we need to make two copies, or like two zippers. One copy goes to one cell, one copy goes to the other cell when that cell divides to make babies. So that process is called DNA replication. This is happening in all of your cells all the time. No, just for time, we're not going to watch that video. Great. So there's a lot of enzymes that are involved in DNA replication. You do need to know the names of these enzymes and what they do, because they all have an important role. I'm going to do a little demonstration with the zipper to help you just kind of like emphasize this fact. But just before we do that, I wanted to introduce the names of these enzymes. So the first is DNA polymerase. So DNA polymerase is an enzyme that actually like makes the DNA polymer. So what DNA polymerase does is it takes those nucleotides, those building blocks, and joins them together to make a polymer. So DNA polymerase is doing condensation reactions. You can think it's like the builder. So it's making that DNA polymer. DNA polymerase is really, really good at doing exactly that one thing. It sees a DNA nucleotide and goes, add another DNA nucleotide, add another DNA nucleotide. That's all it knows how to do. It's not good at starting. It doesn't know, like, where should I start adding DNA nucleotides? There's nothing to add here. So because of that, DNA nucleotide needs a helper, which is called primase. Primase is like the starter. It makes little chunks of sort of tiny pieces of DNA, which are called primers. So these little tiny primers are added onto the DNA before it gets copied, so that it gives DNA polymerase something to grab onto so that it can start adding nucleotides. And then DNA ligase. So DNA, like I said a million times by now, the longest strand of DNA in your cells is 230 million base pairs long. DNA polymerase does not do that all in one long go. That would take forever. Instead, there's like hundreds of DNA polymerase, thousands, all along that DNA molecule, all working at the same time and making like one little chunk. And then at the end, after all those chunks have been made, they have to be glued together to make one nice long continuous piece. And the enzyme that does that glues together those fragments of DNA, those chunks, that enzyme is called DNA ligase. That's like the gluer. So these are the three enzymes you really need to know. So, and then one other thing before I show you the little demonstration, DNA is semi-conservative. So what this means, semi means half, and conservative means the same. So it's half the same. So what that means is that after each round of DNA, what you're going to get at the end is you're going to have two double helices. So you started with one double helix, like 1, 2 strands, right? There's the double helix. And at the end, see how each strand we made is now like half blue and half red. So in other words, the two new strands, or the two new double helices that we make, each double helix is made of one old strand, so one blue strand, and one new strand, one red strand. So that's what we mean by semi-conservative. It's like, instead of like unzipping the whole thing, copying it, making the new strand, like unzipping it and then putting the new strands together and the old strands together, that would be really complicated. Instead, you like unzip the DNA and each old strand is used to make a new strand, and then that's just how you make that new double helix. It's half old, half new. That concept is called semi-conservative. I think the demonstration might help a little bit with understanding what this means. So let me show you this. Okay, so you already have seen multiple times by now my lovely little DNA model. So here's our model of DNA, our double helix. So we're imagining that we are going to replicate this DNA. So I'm going to make 2 zippers. I'm not actually going to make 2 zippers because I only have one, but you're going to imagine with me. Okay, so if I were to draw this, part of what you have to do for your discussion this week is kind of draw this process. So if I were to draw this step right now, what it would look like is it would look something like this. So like here, this would be one strand of DNA, and here is the other strand. And then, sorry, that is not a great marker. And these lines in between, these black lines, those represent those hydrogen bonds between the base pairs. And those are going to break when I unzip the DNA. So this is sort of like our starting state before replication. So this is before replication. This is just the DNA just chilling there in the nucleus. It's going to look like this. Okay, so now the cell is getting ready to copy its DNA. So the first thing it's going to do is it needs to unzip the two strands because it's going to use one old strand as a template to make a new strand. So in order to do that, it needs to unzip these two strands. So to do that, there's this enzyme called helicase that's going to just unzip, go, these two strands, whee, like that. So if I were to draw what that looks like, now that would look something more like this. So this is kind of what the template looks like in your handout for this week. So once again, like this is one old strand of DNA. And then this is the other old strand. Sorry, my markers are not great. And then once again, these little lines, these each represent like one little base pair, one hydrogen bond. So these would be our bases. Okay, so we unzipped the DNA, right? So looking at my DNA again, my zipper here, as I unzip it, that zipper, that is an enzyme called helicase. So I'm going to add to this picture. I'm going to draw, here's my little zipper, my unzipper. That's helping to unzip those two strands. That enzyme that unzips the two strands, that is called helicase. That's how we get to this first kind of step, is we just unzipped. Okay, so now what needs to happen is, remember our goal is to build a new DNA strand. And the enzyme that does that is DNA polymerase. But remember, DNA polymerase is actually really bad at starting, so it needs help from the primer. So what's going to happen is a little primase is going to come along and it's going to start to make some primers, and it's going to use those base pairing rules that we learned before. So G pairs with C, G pairs with C, A pairs with T, etc. So this little tiny chunk, this is called a primer, just a little piece of DNA. And this was made by primase. Okay, so once primase comes in, makes that little chunk, now DNA polymerase, which I'm going to represent as this little thingamabob. Here's my little DNA polymerase. It comes in and says like, oh, hey, cool, there's a primer there. I can grab onto that, and now I can just chug-a-lug, go along, and start copying the DNA. So the way it does that is very similar. It's going to say, okay, what's this first nucleotide here? It's T. Well, what pairs with T? A. So I'm going to put an A here. And then it's going to go to the next one. What pairs with A? T. What pairs with G? C, etc. And it's just going to keep going. It's just going to keep going along. And so that's what DNA polymerase does. It's just going to keep adding those nucleotides, and it's using those base pairing rules to know what nucleotide to add. So how am I going to draw this? Let's go like this, and then I'm just going to like go like that so that you can see. Great. Beautiful. This is the most beautiful thing ever. So this is the new DNA strand that's being made. And that new strand, that is being made by DNA polymerase. DNA polymerase. Can you see that? Yeah. Okay. So DNA polymerase is just going to keep doing that. Helicase might unzip a couple more strands, keep going. It's going to keep making those pieces. But remember I said one polymerase doesn't do all the work. Maybe like another polymerase had already come in and made a couple other pieces. So let's say here's another piece. Okay, here's another little piece. So then the last enzyme you need to know is ligase. Ligase would see these two pieces that were made in chunks. So here's like our two fragments, two DNA fragments. And ligase would come along and join those fragments together. So just join them together. We're joining them. So that's ligase. So it just glues them together to make one nice little continuous piece so that one DNA polymerase doesn't have to do all the work. And that's just going to keep on going until we've reached all the way to the end of the DNA. One thing to note for simplicity, I only showed the bottom strand here. At the same time, some other primase and other DNA polymerase will be on this top strand chugging along and doing the exact same thing. So it is happening in both strands at the same time. I just didn't show that for simplicity. So what you'll end up getting at the end will be one strand that was what we started with before, the zipper in this case, and then the other strand that is all this new stuff I just made. This would be like the new zipper. And that's our new DNA double helix. And that's why this is called semi-conservative, because of this new double helix we made, it's half old and half new. So semi, half, new. So that's our very beautiful little model of DNA replication. I hope that helps a little bit as you are kind of continuing to draw on this type of a picture for your discussion this week. Let me switch my camera back really quick as we finish up the slides. Okay. So quick check for understanding. Which enzyme synthesizes a new DNA polymer by joining nucleotide monomers together? Oh, sorry. So that would be DNA polymerase. So DNA polymerase is the builder. It is the one that joins the nucleotide monomers to each other. Why is it important for primase to make primers during DNA replication? Like pause the video, write out your answer. Okay, so hopefully you said something like primase, it's the starter, right? It has to make those primers to help DNA polymerase start DNA replication. DNA polymerase is kind of dumb. It doesn't know how to start. It only knows how to add. So it needs the starter so that it can just add things onto it. Okay. And then the very last topic, super short, that we're going to finish with is mutations. So this is important because mutations are essentially changes that happen during DNA replication. So if there's a mistake during DNA replication, so if DNA polymerase doesn't add the right base, or if it like accidentally adds an extra base or something like that, that's called a mutation. Because in theory, when we're copying that DNA, it should always be a perfect copy, but nothing's ever perfect. So sometimes mutations happen. And a mutation is just what we consider to be a permanent change in the DNA sequence of a gene. I say permanent because your cell actually has the ability to check for some of these mistakes and then fix them. So if it gets fixed, we wouldn't call it a mutation because it's not permanent. We're just going to watch ever so quickly a video here as well, which I think is just a cute little video to help you understand what are mutations.
The differences in our genes come about through a natural process called mutation. Mutation is extremely common. As a cell copies its DNA before dividing, it makes occasional typos. Most commonly, a single base is substituted for another. Sometimes a base is deleted, or an extra base is added. The cell is able to repair most of these typos, but each round of cell division introduces a few changes that are not repaired. When DNA changes occur in cells that will give rise to eggs or sperm, they can be passed down to offspring. Each one of us has about 60 new variations that were not present in our parents. People commonly use the terms mutant and mutation to describe something undesirable or broken. But mutation is not always bad. Most DNA changes fall in the large areas of the genome that sit between genes, and they usually have no effect. When variations occur in areas of the genome that code for proteins, they often have consequences. These changes can affect the protein product itself, or they can affect when, where, or how much protein is made. Most of the time, mutation generates variations that are neither good nor bad, just different.
Ah, how cute. Not good or bad, just different. That's right. So mutations, I think in our like common knowledge or common usage, we often think of them as like bad things. But really a lot of the times mutations don't do anything. But they can actually be helpful. So sometimes mutations are good, sometimes they're bad. Most of the time they don't do anything. We'll learn a little bit more about how mutations cause changes in your cells, like what the consequence of mutations can be next week. But for this week, just to finish up, you should know a couple different ways that mutations can occur. So the first is one I already said, there can be mistakes that happen during DNA replication. So like DNA polymerase accidentally adds the wrong nucleotide. So instead of like an A going with a T, it goes like A to G. That would be an example of a mutation. There are some things that can make it more likely that DNA polymerase makes a mistake. So for example, the way that sunlight damages your DNA is that the energy from sunlight, so these photons in sun, bind or bombard the nucleate or the DNA molecule itself and actually cause extra bonds to form within that DNA molecule that are abnormal. And that kind of makes the DNA make this like weird kinky shape. And DNA polymerases know how to read that. So it's going to make a mistake because it's like, what the heck is this? I don't know. Let's put a T in there or something. So that is why UV radiation can cause mutations because it's essentially damaging the structure of your DNA. And then similarly, there are also chemicals that can bind to DNA and do similar things that just like make the DNA like a weird shape, and DNA polymerase doesn't know how to read that. So for example, like the chemicals in smoke, there's a lot of carcinogens in smoke that can bind to DNA and just make it have abnormal structure. And then now DNA polymerase can't read it. And so it's going to make mistakes during DNA replication, which if they are permanent are called mutations. Okay, and that is it for module 4. That brought us to the end. So the nice thing about this being a video is if you need to, you can always go back and rewatch parts of it. Please make sure that you have filled out all parts of your handout. And if you have any questions, always reach out to me with an email or you can stop by office hours. Have a good day.